Let's get this underway. Joining us in person for lecture number 2, battling through the difficulties of the modern disease-ridden world. Standing live in front of us for lecture number 2 on stem cell medicine in the modern world, in particular, about using stem cells to develop actual therapies for doing something about actual diseases of blindness. Would you please welcome, huge ISS Thursday afternoon welcome, Dr. Anai Gonzales Cordero.
Thank you very much. My pleasure to be here and, feel this energy. Really good. I hope you enjoyed the lecture last night, but today's going to be different. Today will be fun, at least for me. It's the fun part because I love doing research. And when I decided to study biology, I always knew I wanted to do research and do something that was meaningful. And as I said yesterday, I started with developmental biology, which is basic research, what we call in the lab basic research, understanding the process of biology and life. But then when I started my PhD, I quickly moved into translational research, which is actually developing therapies, doing research that is leading to clinical trials and therapies. And that's what we do, but we also do a lot of basic research because they go hand in hand. So new discoveries need basic research as well. So today, I'm going to talk about the story that we've been through with mainly disease modeling. So I don't know if I can see the pointer, maybe I'll use here. So this is modeling, which is modeling a disease in the dish, and gene therapy, so genetic therapies, and cell therapy. As I mentioned yesterday, my group works in different aspects, all using stem cell technology and organoids. So I will touch a bit on engineering of organoids and a bit on human development, but really what I want to share with you is our journey on the gene therapy and the cell therapy approach. And of course, all in the eye. So let's start by explaining a little bit how the eye forms and the retina, so we can all be in the same page. So from the early blastocysts, which I introduced yesterday, and I spoke about it many times, the inner cell mass, where the stem cells come from, these blastocysts will go through a number of morphological stages, gastrulation, neuralization, and at the neuralization stage, the neural plate starts to form here at the anterior part of the embryo, and that's where the eye field will form. When the eye field starts to develop further, this eye field divides, and then you have two optic vesicles forming that evaginate out of the anterior forebrain that is forming there. This vesicle then again goes through a lot of transformations and invaginates to form the optic cup. And now we have two entities in the retina. We have in the eye, we have the retina here in red, and the RPE, retinal pigmented epithelial cells at the back of your eye. And this is what I'm showing here in this cartoon. I have the neuroretina and the RPE, which is basically, as I said, just this carpet of cells that provides the whole sustentation for your neuroretina, homeostasis of the environment, so on. The retina is then just an extension of your nervous system, your central nervous system, so an extension of your brain, and it is therefore laminated just as your brain is. So we have all these different layers. The ganglion cells that are the ones that will connect to the brain through the optic nerve, the inner nuclear layer with different types of interneurons. So we have bipolar cells, horizontal cells, amacrine, and one type of Muller glia that resides in the inner nuclear layer, one type of glia, which is called the Muller glia. And then we have this amazing layer here, which is the outer nuclear layer, where your rods and cones live. And here I'm showing the rods and cones. So the rod has this elongated specialized cilia that we call the outer segment. So remember the outer segment, because I'm going to talk about the outer segment quite a lot. And this structure has the discs where the opsins live. So the opsins will sense the light. They're responsible for basically your vision. So we have rhodopsins that are specific for rods, and red, green, and blue opsins that reside in the discs of the cone outer segments. And the cone outer segments have a cone shape, and that's why these cells are called cones. Okay, they have different, and I probably will talk about it. Yeah, next. Okay, so these cells have different functions at the back in your retina. So the rods are responsible for peripheral vision and dim light vision, so night kind of vision. And if a patient presents with peripheral loss of vision, such as in retinitis pigmentosa, they will be losing mostly the rod cells, okay? On the contrary, if the patients have loss of central vision, we know it's because they are losing the cone cells that in humans reside mainly in this part of the central part of the retina, which is called the macula, okay? So the macula has your green and red cones, and the peripheral retina has the rods. Of course, we have regions that also have a mix of both populations. But in general, if you're losing the cone cells in the macula, then you're going to have what we call maculopathies or age-related macular degeneration. And in the periphery, you're losing your rods, retinitis pigmentosa. And here's just again a cartoon. You have your healthy retina, and then in the majority of the diseases that are the inherited retinal diseases, we have the loss of the photoreceptor cells. So we have mutations that are causing disease in these cells. And when these cells die, then you lose your vision. There's nothing we can do. The cells don't regenerate, so basically you lost the power of sensing light in the eye. Okay, so of course, if you're now thinking about developing therapies for retinal diseases, we need to understand the stages of disease. And this is very important when you are developing your specific therapy. So you have your healthy retina, normal lamination. At early to mid-stage disease, some of your cones and rods will die, but not all of them. The retina is still relatively healthy. And of course, in advanced stage, pretty much all the cells there have now died, and we are blind. So when you think about translational research and what we can do for these patients, of course, one always thinks first of drugs. You can use drugs to basically slow down the progression of disease, and if you understand what is causing the cell death in the first place, right? What is it that these cells are going through that is causing the loss? But in the retina, we actually don't know a lot about what's happening. We know the mutations, but we don't know the downstream factors of why these cells are dying. And that's because we didn't have a lot of animal models that were good animal models to study these diseases. Another option, relatively new, 20 years worth of research there, and now we're starting to see the first FDA-approved gene therapies going into the clinics. It's genetic therapy. So basically using viral vectors to deliver the gene that is faulty. And if you think about these two approaches, of course, you need the cells there in the first place to receive the treatment, right? So it only works on that early to mid-stage disease stage. Then another two approaches that one can do, and there is another one that I'm not mentioning here, but overall, another two approaches, it's cell therapy and electronic array, what we call the bionic eye. So in this case, of course, they would work, hopefully, in early to mid-stage, but in advanced stage where you lost completely your cells, they are definitely just the two that could work. So electronic arrays are microchips that have electrodes that you could inject at the back of the eye. They will then elicit signals in response to visual signs, and will just, touch the interneurons there and tell them, okay, fire, 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 and send the impulse to the brain and the information to the brain. The other approach is cell therapy, which it's basically the same thing, but now we are replacing the cells that were lost in the first place, so photoreceptor cells. And in a hope that they will connect, so again, the electrodes does need to touch the interneurons. The cell therapy cells, they need to come into this space, figure out where they are, and connect with the interneurons so that the impulse and the light can be sensed. One can argue that the electronic array is a more crude way of doing it. It's a piece of metal at the back of your eye, and in the long term, we don't know what's going to happen with the other interneurons that are theoretically healthy there. And the cell therapy is perhaps a more physiological way of doing it because you actually just replacing with the cells that should be there in the first place. So we took, in the lab, we use cell therapy as our approach, and we also test genetic therapies. And that's what I'm going to tell you about today. As I said to you before, traditionally, we used animal models to understand the diseases of the eye. But they do not always mimic the disease. They can have similar mutations, but the mouse have a very different retina. They don't have a macula in the first place, so to model those diseases that you're losing the cones, it's quite complicated. They are nocturnal animals, so very different from the human eye. So that's where we come back to the induced pluripotent stem cells, because now we have these models where we can get the cells from the patients, get the blood, make iPSCs, derive your organoids, and have thousands of these cells growing in the lab, and I say literally thousands of organoids that we can test our new therapies. And very, very recently, actually, the FDA has approved this Modernization Act 2.0, where they removed the need for animal testing for you to move your therapy to the clinics, to clinical trials. So now they accept models, cellular models, more than before. So organoids are really in the game now. They are the models that you want to be testing. Of course, you can test in monolayers, as I said yesterday. I mean, if you have a good system, why not? But organoids mimic the niche. They are forming all the retinal cell types there. So why not use these models to study disease and then test therapies? Okay, again, just this cartoon, as I said yesterday, but now highlighting the retina, and here, just the outer nuclear layer of an organoid. So this image is showing the cone cells, so the ones that are responsible for your central vision, your color vision, and specifically the red and the green cones that we form in the dish in the organoids. So beautiful cells. They're quite fatty with this nice inner segment here where all the mitochondria are placed. They have this huge process because these cells will form many, many synapses with the other interneurons, and that's what I'm showing in red there. These are all synaptic pantai that we can stain. So indeed, in the organoid, these cells are connecting. So again, it's a very nice model. And as you can see, we are generating quite a few of these cells in the organoid, which is important for us. Okay, so gene therapy, so gene replacement. If you have a faulty gene, you can replace it. And if you want to do cell therapy, you're basically replacing your photoreceptors. Let's start, but before I go into the treatments, what I need to explain is that even though I promote a lot the retinal organoids, and I think they are amazing systems to work with, there are still things that we can do to improve. Okay, it's still a very artificial environment. We are growing a mini retina there, but the thing's missing. So we actively work in the lab to try and improve these models more and more. So we change media formulation, we use bioreactors, we understand development a bit more so we can modulate it further. We want to connect it with other organoids so we form more complex organoids that can maintain the eye environment well. So this is what I'm going to explain to you a little bit in the first part of the lecture, so then when we go into the treatments, you understand that we're using the best model that one can have. So this, it was the first paper that we published using human organoids back in 2017, when I was still in the UK. And this explains basically the protocol that we use to generate the organoids. And I don't think I mentioned it yesterday, so I'll explain this quickly here. It's a very simple protocol, because when I started to test all the different differentiation protocols that are out there, I needed a specific, a couple of things. I needed a cheap protocol, because I knew that I needed to generate quite a few organoids to get cells for transplantation. So I need millions of cells for transplantation. So I need a protocol that it's easy, cheap. I don't need to spend a lot of money with reagents, because everything that has 10 cell names in it is very expensive. And growth factors are expensive. So, and it needed to be something that is not too laborious as well. So as I said, easy and reproducible, robust. This one, it's a mix of two protocols that we tested, and it worked quite well for us. And it's easy because you just let your stem cells grow. Remember that I told you that we need to keep the stem cells at a certain confluence, 60% confluence in the dish, and we split it every time. But I know, and I told you yesterday, that if we leave these cells to overgrow in the dish, they will start telling each other, okay, start differentiating, become something else. And we see that if we don't treat these cells well. So that's basically what we did. We let them grow and touch each other. And when it reached 90 to 100% confluency, we changed the media to a media without two growth factors that are important to maintain pluripotency. And then these cells spontaneously started to differentiate. We gave a little bit of what we call a pro-neural media. So a couple of factors that will induce neuralization, but that's it, not much more, not a lot of growth factors, not much more. It's a very spontaneous process. And we are very lucky because we work in neuroscience, so we are interested in the brain and the eye, and the stem cells actually have the tendency to go towards neural tissues, neuroectoderm. So basically in this protocol, what we see is that after 3 to 4 weeks in culture, and remember, there are monolayers in adherent cultures at this stage, we start seeing these islands of pigmentation. So the brown things that you see there, they're actually retinal pigmented epithelium, RPE, that carpet of cells that I told you that lives at the back of the retina. And from within that island of RPE, we started noticing that we had our neurovesicle coming out of it, a very three-dimensional vesicle that we could see growing out. And it's typical with this bright, transparent epithelia that we call, so a typical neuroepithelia there. So what we do now, and this stage is laborious, and any of my students would say that, you have to sit under the microscope with a needle and isolate these vesicles. And that's when then go to 9/6 plates, and they grow in suspension in 3D as an organoid for weeks and weeks. Here I'm showing a 20 weeks old process, but we actually go up to 33 weeks in culture. So every day, every Monday, Wednesday, and Friday, the guys need to change the media to keep these organoids going for months and months, so they can, at the end, in the 30-week period, get these organoids to study. So what I'm showing here is just one of the figures in that paper where the outer nuclear layer full of rods, so rhodopsin, remember the opsin of the rods, it's here in green, and peripherin, which is another protein that is expressed in the outer segments that senses the light, it's present there. So we were like, yes, fantastic, we have mature photoreceptors, we have photoreceptors that have the outer segments, and that's all we need for them to be functional. Right, and you can see here, this is mitochondria staining, full of mitochondria and other markers and so on. Then we did this technique called electron microscopy, where you can really go very high mag into the cell and see mitochondria. So here you can see typical mitochondria. And we noticed that at the edge of the organoid where the photoreceptors are forming, we also could detect the connecting cilia. So that's the typical morphology of a connecting cilia in the photoreceptor cells. And at the tip of the connecting cilia, we started seeing the outer segments forming. And that was it. And I was so happy when I saw this picture the first time. I mean, I was sitting in that microscope searching for it. Really, it's a huge machine, and you spend all the time in the dark going through it, trying to find stuff. And it's so thin that it's actually hard for you to find things that are not really present in every single cell. And so when I saw this very long outer segments, I was like, yes, fantastic. And you can see there's some discs in there. But we knew that they were disorganized discs. That's not what a normal disc in an adult cell, for example, looks like. So our hypothesis at the time was like, okay, we either have very young outer segments that haven't formed yet, or we have outer segments that are missing that contact with the RPE. So that cell, that carpet of cell that should be there, and in our organoids are not there, they're just here on this bleb of brown cells. So in normal development, these brown cells, the RPE, would be all around it. Nevertheless, good model, we know the photoreceptors form and mature even though they are RPE. Okay, so what we did last year, we published last year, so was to very simply think about the biology and understand what the outer segment needs to form. So it's basically a lipid layer, right? And a lipid layer, if it's forming, needs lipids and glucose to form. The photoreceptors are also very highly metabolic cells. They need glucose to survive and to maintain their own homeostasis. So basically, we supplemented these cultures with more sugar and lipids. And what happened is that now, the majority of the cells, so we have a very significant increase there in cells that have those outer segments. And we showed that now, using the electron microscopy, what we call EM, is that the discs are much more organized. They are discs that look like the discs that we should have in the dish. So now we are happy. We have a model that can generate mature photoreceptors and connecting cilia with proper outer segments. And to model diseases that actually cause mutations in the outer segments, we need proper outer segments. So before we could start any modeling in the dish, we wanted to improve the system, and that's what we did here. Another project that is ongoing in the lab, and Ben is actually, sorry, he's not presenting a poster, that's from the conference. Ben just started his PhD. He's actually finishing his first year now. What Ben is doing, he's trying to develop a very ambitious project, which is develop a macula in the dish. So as I said, human eyes have that central macula, very specialized region, but the organoids don't have that. They're not quite yet there forming a macula. We have lots of cones, but they are not localized in a circle in the center of the organoid. So Ben's job is to now develop a macula in the dish. Why not? So what he's doing is to try and modulate the environment with growth factors such as retinoic acid and FGF8, for example, which are factors that are involved in the cheek in establishing an area that is a rod-free zone. So basically, there are studies in other animal models that shows that these molecules are important for macular development. And indeed, what he's shown is that we actually add quite a lot of retinoic acid, so vitamin A, to our cultures. He decided to remove it. So if he now removes retinoic acid, he sees an increase in the number of cone cells. He's not yet forming a macula, but he's increasing the cells that we need to form in the macula. And also what he notices is that the cells that don't have retinoic acid are actually forming even better outer segments. So for us, it's like, yes, great. And one thing that I removed from here, but it's quite cool that we are doing now, he is going to get a fetal retina, so a very young retina that is developing into the macula. He's going to cryosection that retina very thinly and put it on a slide that has lots of probes, so RNA, messenger RNA probes, okay? And he can then map the central and the peripheral retina, so where the macula is developing versus the periphery, and he will find out every single transcript, so every single gene that is active or off in the center versus the periphery. And we're doing that in a slide, so we have that positional information. Up to now, we could do it in single cells, dissociated cells. But now we have that information, so that's quite cool. It's a relatively new technology called the spatial transcriptome. And Ben is going to now figure out what are the growth factors that we need to generate a macula. And then he's going to go and look at the organoid and see, okay, the organoid is actually missing that. Let's add that factor, or let's remove that factor that is present in the organoid. And that's how he's going to complete his PhD and make a macula in the dish. He would be very happy if he was here listening to me. Because it's tough. It's really tough to do this research because of the age of the organoids, because of the samples that we need. But they're doing a remarkable job. So that's a bit of story on understanding human development and trying to improve the organoid. Next one, it's the work that it's published. So if you guys want to have a look, it's in the stem cell reports. So this is the work, I touched on it yesterday. Remember, brain and retina growing together. So very early on our protocol that I just explained, 2D protocol, we also noticed that we had neuronal cells, other organoids coming out in 3 dimensions. And one of those organoids were brain-like organoids. To be honest, because I was always interested in the eye, we would throw away all these brains. I was like, oh my God, just remove all that crap there. They are really big, and we didn't want it. But when I moved to CMRI, I also established the stem cell and organoid facility, right? So now I had all these researchers coming to me and saying, we need brain, you have neural tissue, blah, blah, blah. And I was like, yeah, okay, of course. I have a 2-in-1 system, buy one, get one free, and you can get my brains out of the retinal cells that we are developing. So, but first, of course, if I'm going to offer my organoids to anyone, I need to characterize the organoids. So that's what we did. We collect the brains, and we figure out that they are actually dorsal cortical, not ventral, dorsal cortical parts, brain. And again, you can see the rosettes that I showed you yesterday. They are there. They are functional, so if you put them on the microelectrode array, they will fire. They are good organoids to test. But then another interesting fact is that we would see that sometimes we had brain and retina close together, and now instead of separating it and using the retinal organoids to understand retinal disease and the brain to understand brain disease, we kind of culture them together. And here you can see, so this is an image, one after the other. So when we cryosection these organoids, it's a very thin, 14 microns worth of tissue that we cut in a very cold, so -20 chamber. And we collect all the sections, one after the other. And this is what you're seeing here, is a progression going through the brain organoid on this side and the retina on this side. And you can see this structure that is crossing over and actually forming a nerve band-like structure coming from the retina. And I know it's coming from the retina because in red I have a marker that it's present in the retinal ganglion cells only. So these are the retinal ganglion cells that are forming this nerve, this bundle of nerve-like structures, and they are sending it towards the brain. And you can see, if we look at the brain here on the other side, now we have retinal ganglion cells, axons that have reached all the way to the brain. And this is literally what happens in normal development when you have your optic nerve going towards your brain. I'm not saying we have an optic nerve here. We have a nerve-like structure that is trying to connect with the brain very early on in culture. So this is the week 10. So we do need to now grow it further and see how these 2 complex organoids form together. And here is just a video. Hopefully it's going to play. Yeah. So we're going through the retinal organoid. okay? And in gray, you're gonna see very soon the retinal ganglion cells that are labeled, so there they come with the processes. And if you look carefully here in the middle, see the nerve forming, going all the way through, and that's a bit of brain there. Okay? And neuroepithelium of the retina with the ganglion cells, and they send it all the way there. So quite cool that we could visualize that in a video like that. So it's all published, and have a look. So this is our shot at doing a little bit of engineering and trying to grow these organoids together. Next, we developed this retinal atlas. So, of course, if you think about therapies and you want to transplant photoreceptor cells that we are making in the dish, the best that we can make, you need to understand how similar these cells are from the real human photoreceptors from the adult retina or the fetal retina, right? How bona fide these cells are. And basically, this is the study. We used computational biology and a lot of data sets that are out there that have been used to characterize individual cells, not only in the organoids. So this is data sets from a number of organoids from different labs. So this is called single-cell RNA sequencing. So basically, you can mush it up your organoid, dissociate it, and you can then read the transcripts, again, messenger RNA, from different cells that were present in your organoid. And you form these clusters of different cells. So you know that the pink ones will be the cones, the blue ones will be the rods, and you assess then based on the clusters and numbers and cell proportion in this data set. So what we did here, we used a statistic program that was developed by Honey to compare a number of human data sets with our organoids. And our organoids actually rank quite well when you compare with other protocols out there. So I was very happy to see that. And they're right in between. So these were 30 weeks old organoids, so almost birth kind of organoids. And they're right in between the fetal and the adult tissue, which was quite cool because we could replicate that in the dish. And very, very similar indeed to the human, the bona fide cell. So good. Again, this is published. The paper's out there if you want to have a look. So we have cells that are very good to work with. Let's get into disease modeling. I'm a bit slow today. And what we call disease pathophysiology. So understanding the disease in the dish, basically. So the majority of the ciliopathy, the diseases of photoreceptors are ciliopathies. We have a number of diseases, actually, from mitochondria defects, phototransduction, which is the phototransduction that happens in your outer segments, IO channel defects, and so on. But the majority are cilia proteins. And one of the ones that I'm going to explain that it's actually a ciliopathy. And again, here, just to emphasize that we have a connecting cilia and the segments in the photoreceptor cells. So two very important structures there. I also need to, before I move on to explain a little bit more about disease, I need to explain gene therapy. And I'm sure you guys know about it, but just in case, we use the vector, a virus, as a vector. So basically, it's a delivery truck. So we modify this vector, this virus, to insert a piece of gene there, whatever gene we want, a gene of interest. And once we insert this gene, we can then, of course, in vitro, we package this gene inside. We have a helper plasmid. We have a whole array of 2 weeks worth of experiments that we do to make these viral vectors and purify it. So we have good vectors to use in our cells of interest. But basically what happens is that this vector then attaches to the cell membrane, gets internalized, and then it goes to the nucleus, and we have the gene of interest being transcribed and then translated. Okay? So it's just a delivery system. And what we use, and is safe to use in the clinics, and again, we have actually an FDA-approved treatment. Now we have 3 or 4, actually. They're approving more and more, but we have one for eye. It's the adeno-associated viral vector. So they are, we call them capsids, and they're viral particles that have been modified so they don't proliferate, they're not dangerous. They, again, just are, are a delivery system. But there is a caveat to this amazing system. It only fits 5 kilobase worth of cDNA there of your gene. So it's a good size, and there's quite a few diseases that we can actually replace the gene with 5 kb, but it's not optimal. How do we use gene therapy in the eye? So in the eye, we're actually quite lucky because the eye is on the outside. We can access it easily. We actually have quite a few treatments that involve injections into the eye, into the vitreous cavity here. So basically, you go through the front of the eye and you deliver your drug or your viral vector, right? But this viral vector, if delivered to the vitreous here, will only touch these ganglion cells and the inner nuclear layer. Because right here, between the ganglion cells and the inner nuclear layer, you have a membrane where this vector cannot cross, okay? Sometimes they will go and reach the photoreceptor, so it's not that bad, but it's not an optimal. So another delivery method, it's subretinal. It's much more tricky and difficult to do. There's only a few surgeons out there that can do this surgery, and it's the delivery of the vector in the subretinal space. And this is important space that you need to know about because that's where we're going to deliver our cells as well. So it's right in between the retinal pigmented epithelium, again, the carpet of cells, and the photoreceptor cells. It's not a natural space that you have at the back of the eye. It's just a pocket that we create when we inject. So the photoreceptors are touching your RPE daily. The RPE actually has a lot of microvilli that needs to phagocytose your outer segments. So the outer segments that you produce actually are shedded daily so that they can be replaced with new opsins. And that shedding requires, you need to get that rubbish out somewhere. So that what happens is that the RPE phagocytoses all those outer segments and does all the wonderful stuff as well, but mainly phagocytoses the outer segments and clears up the environment. What we do, we just lightly lift the whole system and deliver what we want in there. And again, as I said, there is one FDA-approved treatment for LCA due to mutations in the RPE65 gene, which is actually a gene that is mutated in the RPE cells, okay, and not in the photoreceptor cells. Anyway, moving on, how about large genes? I mentioned to you, yes, we can treat diseases that are due to mutations in genes that are up to 5 kbs. That's what we call standard gene therapy, and I'm not going to go into that today. We have a greater challenge in my lab. What we want to do is to treat larger genes. So how about the ones that are large, very large sometimes? We can do two things and more. There's other technologies out there, but the two things that we do in the lab is to use a dual AAV system, which is very simple actually. Now, instead of using one viral vector, we're using 2 viral vectors carrying each. They will have half of your gene. So we'll cut the gene half in the middle, and one viral vector will have half, and the other viral vector will have the other half. And then once in the cell, they have a piece of DNA that recombines, and hopefully then the transcript can be made entirely, and then you have your protein. Okay? But if you think about it, to get 2 viral vectors inside the same cell is not as efficient as getting one. So we're testing it, and we'll figure out very soon if it's going to work on our organoids, but this is ongoing, and I'm going to mention it in a bit. Another process to use viral vectors to, how I'm doing for time? Yeah, okay. Another approach is to use CRISPR-Cas, right? So, and use the AAV, the viral vectors, as a delivery truck for our CRISPR-Cas machinery. So what is CRISPR-Cas? And I know you guys have heard of genomic engineering and CRISPR, but just to quickly explain, so it's clustered, regularly interspaced, short palindromic repeats, CRISPR for short, and we use a nuclease called Cas9. There's a number of nucleases out there now, But the main one, the first one discovered was Cas9, and basically it cuts your DNA. So it makes double-strand breaks in your DNA so that the DNA can be altered. But it needs this guide RNA sequence, so it's a very short 20 to 21 base of pairs, nucleotides that have this PUN sequence that can guide the Cas9 nuclease to the right place in your genome. So it's very precise. It goes to the right place on the genome where you want to alter. And you can have two types of alterations. You can have non-homologous end joining, which is basically, it's going to go there and cut and form what we call indels. Basically, it's just going to mess up with the whole sequence that is out there. Cut, 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 makes different cuts and mutations in that region and can cause frame shifts or loss of function of that gene because you're just kind of messing up with the region. And then you can have homologous direct repair where you have a donor template that will use as a template to then correct the region that is faulty. The problem with homologous direct repair is that it doesn't work in post-mitotic cells, so cells that are already mature and are not proliferating anymore. So to use HDI in photoreceptor cells, it's very inefficient. We can't really do that. But anyway, there's other ways that we can do it nowadays. So how we do it in the organoids. Of course, we know a lot about, and I was in a lab that did a lot of gene therapy. Actually, they were the pioneers in gene therapy in the eye. They did all the development of the RP65 trial. You know, they were very good in gene therapy, and they knew all about gene therapy in the eye in rodents, and then they did lots of clinical trials. But when I started thinking, okay, let's use organoids and let's treat the organoids, we didn't know anything. I mean, how much are we going to add of the vectors? When are we going to add it? What is the best? Because viral vectors come with different flavors. They have different capsids that will have different infectability to different cells. So what is the best one to use in vitro, and so on? So we figured that all out, and that's a summary of one of the projects that we published in Human Gene Therapy, where what I did was basically to screen all the main capsids, so all the main flavors of AAVs that are out there, and they have these funny names, AAV2. They're all based on AAV2, which was the first one, but AAV29, AAV25, and that means different mutations in their capsid, so they have different abilities to, different tropism to different cells. So at the time, in this study, we found that this SSH10 capsid was the best one in vitro to use, not only on organoids, but also on RPE cells, in mouse cells, and human organoids, and so on. Later on came 7M8, another type, and it's even better than SSH10. And now what we have in the lab, it's actually novel capsids. At CMRI, we have a group that studies just development of novel capsids. So basically, they engineer the capsids, they do direct evolution to change these capsids all the time, so we have new capsids that are better and better. And of course, another thing that we can do is to use different promoters. So promoter is a sequence at the front of your gene that tells the gene to be expressed in a certain way. So a promoter for a cone would be expressed just in a cone cell. It's driving a gene specifically expressed in the cone cells and so on. So you can have AAVs that, although will go into all the cells, they'll be on just in a specific cell depending on the promoter that we use. And I hope it's all clear, guys. Well, we'll have questions later. Did I miss anything here? So no. Okay, one thing that I need to mention is, of course, if you want to do disease modeling, you need to find a biomarker of disease. Of course, you know how to do your gene therapy in vitro, but you also need to find a biomarker, a disease. We need to make the organoid have a disease characteristic. Otherwise, I mean, how are we going to rescue that? So the two conditions that I study are both due to autosomal recessive mutations in large genes. And they're the most common inherited retinal diseases. And they don't have any treatments because they cannot have the standard AAV gene therapy that is out there. And these are two, so who, local people know that CMRI, I mean, we have a campaign every year called Genes for Genes, and not everyone actually associates Genes for Genes with CMRI, but we've been doing it for many, many years now. And every year we have a face. The kids are the face of Genes for Genes, and these are two of our patients. And Arato, he actually has Stargardt's disease, and these two boys have Usher disease. And these are the conditions that I'm trying to develop treatment for. So Stargardt's, it's a retinal degeneration, it's a maculopathy. So he's going to lose the cone cells in the macula, and mainly around the second decade of life. Usher, it's very devastating. It causes deafness as well as blindness. And depending on the subtype of Usher's, you can be born deaf, or it develops as in early childhood. Blindness also develops a bit later, but again, it depends on the subtype and the severity. We don't have any treatments, and the standard gene therapy is not an option. So what are we doing in the lab? Okay, here just explaining a little bit more. It's a bit of a busy slide, so I don't like it too much, but Stargardt's, it's localized to the outer segment, so that's why we needed good outer segments to study this disease, because it's basically a protein that flips information into the outer segments, okay? When these outer segments, when this protein is mutated and this piece of outer segment is engulfed, phagocytosed by your RPE cell, because this flipase is not working, we have accumulation of this A2PE compound in the RPE cells. It is toxic for the RPE cells, so then accumulates these deposits there, and the RPE cells die. And if you don't have RPE cells to provide sustentation to your photoreceptors will die. So that's how the disease occurs. It's actually death of RPE and then photoreceptors. That's what the field believes. Usher2A, and here I'm concentrating on a subtype of Usher, which is Usher2A, which again is the most common. Within this Usher syndrome subtypes, Usher2A is the most common, and it's due to mutation in the Usher2A gene, 15 kilobases, so really big. And encodes the transmembrane protein here at the base of the outer segment and the cilia, and encodes this protein called ushering. And really, that's all people know about Usher2A. They know where the protein is, but they really don't know what this protein is. The hypothesis that is there is a big transmembrane protein and is there holding your cilia and the outer segment together in place so everything can work properly. So what we did in the lab, we generated iPSCs from 3 patients from each of these diseases. We can grow the organoids. And important that you see here is that on the outside of the organoid, you see this hair-like structure. I call it the hairy organoid. It's all full of outer segments. These are our brush border, full of outer segments. And of course, if you want to study a gene and a disease, you need to have the protein present for that gene. And here, the first thing that we did was to show that ABCA4, which is the gene that is mutated in Stargardt, it's present in the outer segments of our organoid. So great, we can model the disease. For Usher2A, same thing. We checked good organoids, and we checked that ushering was present, and it is. So, okay, what did we do to now start modeling this disease? So Mel is a postdoc in the lab working on it. She started to develop the organoids, and here, very simple, all I'm going to show is that our diseased organoids, so the Stargardt's organoids, now no longer express ABCA4. They have a little bit of it, but not enough. So the disease is going to start to develop. We confirmed that using proteome, and then we did single-cell RNA sequencing. So now we can isolate all the different cells and check for the transcriptome on specific cells, right? And surprise, what we saw was that we have a reduction in our cone cells. So remember, we don't have RPE here. So it's not that the RPE is dying and causing the death of cones. we have a reduction in cones without the presence of RPE. So there is some kind of toxicity going on there in our cones. And we confirm that with other markers and proteome as well. So this is the beginning of us trying to find a phenotype in the dish. And then what Mel is doing now is to do that dual AV system. So the ABCA4 gene is 6.8 kb, so it's almost there, could almost fit in one, but not enough. So what we did, we cut the gene in the middle, so we have exon 1 to 24 in the left vector, and 25 to 50 on the right vector. And as I said, we have these donor sites and recombination signatures there. So once inside, this DNA can recombine and hopefully will be transcribed into full-length protein. And this is what Mel is doing at the moment. And here I'm just showing that there is an upregulation of ABCA4 after we use the gene therapy in our organoids. So we're in the right path to hopefully produce fully functional ABCA4 protein again, and hopefully make these photoreceptors healthy again. Okay, so that's the Stargardts. Now, Usher2A. Usher2A is different. The cell lines that we have this deep intronic mutation. So, as you know, we have introns and exons. The exons transcribe your genes, and then the introns are actually sequences that are there, and you don't really need it to make a gene, right? So mutations deep in the intron, in this case, this mutation will cause a frame shift that will have a stop codon, so your protein will stop a few base of pairs later, because it's basically an A to G mutation, and this will start to be read differently, and a stop codon mutation will appear somewhere down the line. And what happens here is now we have the formation of this pseudoexon. So we have a mini exon that is formed because of this mutation. And we can actually see it here with this extra band on the gel. This is the wild type ushering, and this is the extra only on the patient samples. So the idea here is now to use CRISPR-Cas to go in there and cut out this A to G mutation. So we get our Cas9 to go exactly where this mutation is with our guide RNAs that we developed. We have it here. And yeah, and cut around this mutation. So we're actually using two guides. We have one that is before the mutation, one that is after the mutation, so that we create more mass and hopefully remove this region, and we don't have the formation of the pseudoexon. This only requires one AAV vector because we're using a Cas9 that is small, and everything fits into 5 kb of a single vector. And I mean, a lot of data here, but the bottom line is that now, with the guides that we developed, we understand much more how the CRISPR-Cas system works in organoids. So we know that at 2 weeks after, so if we use the vectors and we collect and treat, and then we collect the organoids 2 weeks later and assess to see if the DNA has been cut or not. So this is cutting efficiency of your DNA. 2 weeks, not much is happening, so around 4%, which is not great. 4 weeks, that increases to 10%, and 6 weeks, now we're reaching up to 20%. So we're improving, so definitely the CRISPR-Cas system needs more time inside the cell to cut, and to create indels there. We're also testing 2 different capsids, again, the 7M8 and this M4, which is the novel one that we generated in the lab. And very nice to see that the new one that we generated actually cuts better than the 7M8. And we're starting to see a reduction of this extra band there on our gels. So indeed, now we have more wild-type ushering versus the pseudoexon that is causing the disease. And that's all we need, because hopefully then the cell will be healthy. So very brief, going through this work that Deborah is doing, really good postdoc in the lab. Anyway, when you talk about ushers, I also said that ushers cause deafness, right? And we have another student now, Vivienne, that just started, and she's doing inner ear organoids, which are a different game. You can see here how beautiful they are. They have otic vesicles full of hair cells. So this is a sensory epithelium with the hair cells. This is a highlight of one hair cell that is in your vestibular system. And here's the hair bundle that senses the mechanical, the movement in your inner ear. Very hard to generate inner ear organoids in comparison with retina. So she's doing an amazing job. Anyway, cell therapy. This is the fun part, and I know it's been a long day. So how many?
In 5 minutes,
they're going to have to leave. Okay.
That doesn't mean that you've only got 5 minutes. We'll just need to take a pause.
Yeah. Okay. Guys, this is the most exciting part to me because I've been working on it for so long. So it's transplantation of healthy cells to rescue vision. Now, I'm not talking about diseased organoids. It's cells that come from healthy individuals that we generated iPSCs from, or we use embryonic stem cells as well, have the organoids growing, isolate these cells to transplant, and hopefully rescue vision. And that's the story that I'm going to tell you about. So we started this work. This work started many, many years ago. And the hypothesis is the eye, it's very easy to do transplantation. So it's a very feasible space to start doing cell therapy because, again, it's accessible. So you can just inject. And very early on, we learned how to transplant and deliver gene or cells at the back of the eye in the subretinal space where we needed it. So basically, if you're losing your photoreceptor cells there, and you just come in with a new population, this population of photoreceptor cells has to do two things only. One synaptic connection with your next cells in the inner nuclear layer, your bipolar cells, and they need to form outer segments to sense the light and be functional, okay? So the eye is a simple structure, is accessible, and there is one-to-one cell synaptic connection, and the cells need to mature. So if you compare this with the brain, where you're transplanting, let's say you're transplanting dopaminergic neurons to treat Parkinson's, those cells actually have to connect with lots and lots of other neurons, and they actually need to send nerves all the way to other places. So it's a big task. In the eye, actually, we're doing, relatively well, and it's easy. Okay? So we started this journey back in 2006 when we transplanted mouse cells. So these were healthy mice that were very early in development. So first week after birth, we had a marker that was specific for photoreceptor cells, so green fluorescent protein expressed in your rod cells. And we isolate these cells, and we transplanted them for the first time, showing that transplantation was possible. I then moved in and a lot of work happened in between. In 2000, I'm not showing it here, but we had another paper a few years later, after 2016, I can't remember when this one was, 2012, restoration of vision. So basically now we optimize this system and I probably have it later. We optimized this system enough to show that we could rescue vision in blind animals. When I came into the game in 2013 to do my PhD, I started using stem cells because getting primary cells from mouse is one thing, and if you want to do that with human fetal cells, which was being done, people were trying to isolate retinal progenitor cells and photoreceptors from fetal tissue to transplant, it's actually quite hard. I mean, there's not enough tissue out there to do this type of study. So we needed a renewable source of cells, and that's when stem cells came into play. That's when Sasai started to develop the optic cups, and everything came together for us to be able to get enough photoreceptor cells. And I did first in the mouse, and that's the work that I published out of my PhD. And then we did using human cells human photoreceptor cells in 2017 when we published our protocol. Very recently, we showed, so that's the 2017. Very recent, 2021, we showed that using the human cells, we were able to restore visual perception in blind animals. And that's what I'm going to show you today. This is the very first 2016 paper in Nature where I just... where I just show the actual cells that we transplant. As I said, we had these fluorescent, green fluorescent cells, rod precursors from postnatal day 1 to 8. Actually, we found that that was slightly better. We can isolate these cells, transplant at the back of the eye. At the time, we were transplanting 200,000 cells, and you can see the cell mass there at the back of the eye. And it was the first time that one of the first times that transplantation was possible. So retinal repair using photoreceptor precursors. And the beauty of this study was, let me see, I don't have that here. Is this now not working? The beauty of this study was that we figured out that the stage of the cells is important for transplantation. So what we did is we transplanted embryonic cells, so earlier than postnatal, so embryo-like, postnatal, and adult cells. Embryonic cells, they loved the subretinal space. They were there, and we could see the cell mass, but they started to form their own niches in the subretinal space. So instead of connecting and going into the eye, they started to form rosettes and connect to each other. So no good. What we need is these cells to migrate into the outer nuclear layer and to connect. The adult cells, the same thing. They couldn't go in and connect because they're too mature and probably didn't have the plasticity to do it. We were removing the outer segments that were already formed because when we dissociate these cells, to go through a machine, and I'll show you. So basically, we use FACS sorting. So it's a machine that has a laser that can detect this green fluorescent protein and separate the non-green with the green cells, and we can then transplant a purified population of cells. So when we have to do the manual dissociation, these cells lose the outer segments. And then somehow the old ones didn't like it and were not functioning properly. But the postnatal ones that were actually precursors, they don't have the outer segments yet, did quite well, and they were able to migrate into the layer and connect. At the time, we couldn't get a lot of functionality out of these cells, but as I said, 6 years later, in 2012, we optimized the system so that now we had a lot more cells going in, and we were able to do a lot of behavior studies to show that these cells are actually functional and are restoring vision. So we did this water maze study. For example, we had to train the animals to swim, and they had a computer screen that there was a signal. So they had bars, and if the animal could see, they would escape to a platform because they don't want to be in the water, they don't want to swim. So the stimulus there, the light stimulus, was the rewards, the platform. So they quickly learned that to assimilate one to the other. And of course, the blind animals couldn't do that. They would just go around searching, and then by chance they would end up in the right side. But the ones that can see would go to the platform with the light. And we correlated that with number of cells in the subretinal space, with the ones that could swim and find the reward. We also have this optokinetic optomotor machine. Basically, it's a 4-screen chamber where we run, again, these stripes at different light intensities and different thicknesses, and the animal naturally follows this. If you run this drum around, this stimulus around, the animal will follow, like if you are in a train and the train is really fast and you see the movement, you end up following it just to see what you want, what is happening outside. And with the animals, it's very clear they follow the line. So that's how we can measure visual acuity. and they performed better when they got the cells. We also could see responses in the brain, all the way in the visual cortex. So it's very, very nice to see that transplantation of cells was restoring visual function there. And something that I'm not showing here is that later on, we discovered that not all the cells that we transplant actually migrate. And one thing that I don't show nowadays anymore is actually the image of the cells inside the layer, which is so important for you guys to visualize. But you see one with the stem cells in a minute. So what happens is that we discovered that not all the cells would actively migrate into the layer and connect, but the cell mass that was there was providing vesicles and would connect with the other cells, not necessarily through a synapses, but just through nanotubules, and send proteins or messenger RNA to these endogenous cells and rescue vision that way. So there's two mechanisms. One is that active migration and connection, and the other one is basically just replacing the protein through the connections. So the cell mass is basically just delivering whatever is missing in the animal model. Both valid, because both we could rescue vision there. But that's with mouse cells into mouse cells, right? How about pluripotent stem cells, and can we generate enough cells to do these experiments with a renewable source of cells, pluripotent stem cells? And as I said, that's what I did during my PhD. I actually have another publication before this one where I was doing the 2D protocol, 12 hours a day, in a safety cabinet growing cells, crazy stuff, to get like 1 vial worth of transplantation. And we transplanted, but these cells were not good to integrate into the retina. So they would sit in the subretinal space, but not do much. And at the time, we're like, it's because the cells are not good enough. We knew we needed that specific stage in development, right? We need that postnatal stage. We were getting something that was before that, because the 2D environment wouldn't allow the development of the cells. But there came Sasais and Uraku that did this wonderful study that I showed yesterday with the optic cups. We quickly replicated that study, and we generated the embryonic bodies and the vesicles. The problem is that if I wanted to sit down there and cut the optic vesicles out of the organoids, I wouldn't get anywhere, I wouldn't get enough cells, and I wouldn't have the time to do that. So we optimized the protocol so that the vesicles would stay intact in the organoid. So that's the organoid with the 2 eye vesicles poking out. And we manipulate the system so that they would survive and mature without us having to cut it out. So for us, it was a game changer, because now we could have lots and lots of organoids growing and generating photoreceptors. And here what we did was, we didn't have a line, a stem cell line, a mouse embryonic stem cell line that had our photoreceptors expressing a fluorescent protein. So because in the lab we had access to gene therapy, what we did, we used AAVs now just to highlight our photoreceptor cells. So I used an AAV9 vector that at the time was good in retinal organoids with a rhodopsin promoter. So I'm highlighting here just the rod cells driving a GFP, so green fluorescent protein. And you can see that now the organoids have quite a lot of vesicles full of rods. And here's just a cross-section through the organoid. Sorry. And then when you run it through the fax, again, so that's the fax sorting machine. It has lasers that can detect the green cells, and it's basically droplets. It's going through droplets and sees the green fluorescent protein and says, okay, you go there, and then the ones that are negative go to, and it basically collecting tubes. So you're purifying your population. I did that, 25% of my cells were green, and I transplanted 200,000 cells at the back of the eye. And here I'm showing the cell mass, so all the rods in the subretinal space. We were using this rod transducing knockout mice, so these rods don't express, they don't have rods transducing this protein, and what happens is that they have night blindness. So basically, they are still present. You can see that the outer nuclear layer, so that's where all the photoreceptors are, is still quite intact and well, but the rods are non-functional because they don't have rod transducing, which is necessary for your phototransduction cascade. But you can see that the green cells that actually migrated, so this is what I should have shown you for the other one, the cells that go and migrate, now express in red there rod transducing. So we know that these cells are actually embryonic stem cell derived rods. And this is just a highlight, just highlighting that. And rod transducing is actually expressed in the segments where it should be. Where we also looked for the other structure that we need for maturation, which is synaptic connection. And that's what we saw, is that our rods had beautiful axonal buttons there, connect very close precisions with your bipolar cells. And when we started to stain for synaptic markers, such as rebi, so all this puntated staining, it's synapses there. The synapses are present in the axonal bundle. Now, at this time, very few cells were actually integrating out of the 200,000 cells. So we didn't try to do any functional assays. We knew we were going to fail. So it was just proof of concept that mouse embryonic stem cells could be differentiated to a stage where integration and this migration into the outer nuclear layer is possible. What happened next is that I started to work with human pluripotent stem cells because I knew, okay, we need to go from mouse to human. So is that stepwise? We first use the mouse into mouse from primary cells, then we move into mouse embryonic stem cells, and now how about human pluripotent stem cells? And that's when we developed the protocol that I already introduced, easy, cheap, and efficient, because we need thousands and thousands of cells. You can see that your photoreceptor cells are on the outside of the organoid, lots of them. The first thing that we did was to actually characterize these cells at different stages of development and figure out which one was the stage that we needed for transplantation, similar to the rodent. And we transplant now typically at 17 weeks of age, so it's still 120 days that we have to grow these organoids for, but it's better than 30 weeks that we use for disease modeling. At the time, we used a hemopsin promoter, so now we're talking about cone cells because it's human cells, so we were interested in actually trying to replace cones and not rods. And we used an hemopsin promoter. Now we're using the SSH10 capsid because we knew it was better. And we had 7% of the cells green. We transplanted, and you can see the human cells. So this is a cross-section of the eye, almost the whole eye, and you can see the spread of the cell mass throughout the eye there. Very happy, because this was a mouse eye, no immune suppressed, receiving human cells. Then one thing that I need to show you here is that this was the wild type with the cell mass, and this is this model here, AAPL1. So it's an advanced degeneration model. Remember that I mentioned that for cell therapy, we actually want to be treating the more advanced cases. So this is what this model mimics. It's lost all its photoreceptor cells, so there's no outer nuclear layer. So this, where the photoreceptors are, is not there anymore by 1 month. So it's a very early rapid degeneration, and all we can do is to transplant the cells back. Now, it's also important because we know that there's no photoreceptor cells there, so the method that I just explained before of transfer of material from the cell mass to the endogenous cells cannot happen because the endogenous photoreceptor cells are not there anymore. So any rescue here, it's coming from the cells that we transplanted because there's no mouse photoreceptor cells left. So at this stage, all we did was to see a few human cells, and I know it's a human cell because we have a human nuclei marker, and you can see that the nuclei of the cell is actually quite large in the human cell, and because this is a cone, they're quite large cells, they're quite nice. And a few of them started to go into the right layer and kind of send process to try and connect. Here in the AAPL1 model, where the outer nuclear layer is gone, we see that all the cells sit there, form a semi-organized layer that tries and connects with the endogenous retina. And again, that rebinds, so the synaptic marker, punctated, is expressed there. But again, very few cells, and we asked the question, are these cells connecting? At that time, we didn't know. And then what happens is that I have a and I don't have her picture, but it's a very talented PhD student, Joanna, that optimized the whole transplantation system. So here we thought, okay, we're transplanting 200,000 cells, but normally in the mouse retina, we have more like 3 million cells or more. Can we go higher? Can we transplant more cells? So she increased to 500,000 cells, so 500,000. The problem here is that, of course, we were transplanting human cells into the mouse, and it would lead to rejection. So what she did, she crossed her advanced degeneration model with a nude mice that doesn't have thymus and then therefore is immune compromised, so that we could keep the cells in the subretinal space for 12 weeks now, so 3 months after transplantation. Before, the work that I was doing was all 4 weeks, 1 month. So, and we know that the human cells need more time to develop and mature. So 12 weeks was a good time point. We also had a really nice control, which was some cone cells that we generated from organoids that have this disease, CNGB3 disease, where the cone cells developed and they are normal, but they are non-functional. So our best control was normal-looking cones that are non-functional. Okay, so what Joanna did was to produce lots and lots and lots of these organoids. She purified the population, and she transplanted. So this is the beautiful image at the back of the eye of that rodent with all the cone cells there. I mean, a really nice cell layer, not organized, but nice. And in gray and white there, you see the bipolar cells, which are the ones that need to connect with the photoreceptor cells. And actually, if you look carefully, they are sending processes towards the cell. So there is some kind of connectivity happening there. Oh, there is Joanna. Good. And what we did next was to try and now assess if we can rescue vision. So she isolated the retina. So after transplantation, after 12 weeks, she removed the eye, cut just the retina out, and she placed it on the microelectrode array that I introduced yesterday. So all these bars here are electrodes in the array. And in green is the area where the cell mass is. We could see, because it was so bright and nice, that we could see where the cell mass was. And she put it the other way around, because the cells that spike in the retina are actually the ganglion cells. So we transplant the photoreceptors, they need to connect with the bipolar cells, and the bipolar cells need to connect to the ganglion cells that fire in response to light, okay? So only if there is that complete connectivity, we are going to see the ganglion cells firing in response to light. And I know that because if we do a blind mice, an untreated blind mice, there's no response to light. So light shines here at 0. And the only thing that we see is these very prolonged tiny bumps. You can see no peaks, right? Now, on the ones that we treated with the cones, where the cell mass is, and in close proximity, you can see that time of light, 0. You have this very rapid response, shoom. So that's the spike of the cell. In this case, I'm also showing ERG, a microelectrode of the actual cone cell that we transplanted. And then we also tested drugs. So if you can add drugs that inhibit specifically synapses of the photoreceptor cells, and we see that we abolish that response. And then when we wash the drug out, then we actually have the response again. So these are all controls that we need to do. And on the animals that were treated with the CNGB, importantly, again, we didn't see any of this rapid response. So cones respond very rapid, and that's why I say, it's because that's typical of cones. We saw this prolonged bump again. And now we know that these are actually melanopsin positive cells, so these are ganglion cells that respond to light because they have melanopsin, but it's this very different response, which is prolonged and bumped. So we here specifically got these ones just to show as an example that there will be some cells that respond this way, but it's not the cone response that, because these cells are non-functional. We also did behavior studies. So in this case, it's, we have two chambers, one that is dark and the other one that is light. Mouse are nocturnal animals, so they prefer to spend their time in the dark. So a wild type animal here in black with the open circle spend the majority of the time in the dark. The blind mice, they don't care, they don't see, they spend 50-50. Now the transplanted ones in green now started to spend more time in the dark, just as the wild type, and the one that received the shunt cells, again, they don't care where they are. And this is depicted here. So these traces are us tracing. So we have a camera at the top of the arena, and we can trace and see where they have been, and we can see where they freeze. And basically, bottom line is that with the transplanted cells, these animals acquired more of a behavior of the wild type animal that can see properly. So that was really good. We showed that we can rescue some kind of light perception after transplantation. And this is amazing because now the group in London is actually developing GMP cells, or good manufacturing practice cells, to try and move this work into clinical trials. And we are doing the same here in Australia. So next, what is next? When I came down to seasoning, I was like, okay, we've done cell therapy, it's wonderful, it works, but we need to improve it. It's not good enough. So lots of different ideas. How can we improve? One, yes.
Let's check. I just want to make sure that we do have some time for questions.
Yes. So one idea, and I'm just going to present this quickly because I know about I've been just so slow.
Sorry, I don't want to rush.
People can modulate the environment. So we want to modulate the environment to promote more of that connectivity, right? That's basically what we need. We need more synapses to form after transplantation. So you can use signaling molecules, you can use biomaterials, perhaps transplant your cells with extracellular matrices, blah, blah, blah. We actually went for a very different approach, which is bioelectrical modulation. So we want to stimulate the cells after transplantation, before and after, with electrical stimulation to promote axonal migration and connectivity. And this is, has been tried in clinical trials for people with retinitis pigmentosa, and actually it has been shown that it slows down the disease progression. So your photoreceptor cells don't die as much if they keep firing and keep active. So basically this is a very biomimetic electrical stimulation at the front of the eye. You can see the device is nothing fancy. It just literally delivers some very gentle stimulation at the front of the eye that will have an effect in your retina and just keeps the cells ticking, you know. The other cells that are there, it promotes these ions, these receptors, to be there in the right place and work. So what Michelle did was to use electrical stimulation in vitro in her organoids, and she showed that she can mature the organoids more, and in fact produce more photoreceptor cells when we deliver electrical stimulation in vitro, which is quite cool because we need more cells for transplantation. And when she did it in the animals, so this is the nude animal that she uses, no coat, and is immune compromised. We did this using a contact lens with a gold electrode. So she transplants the cells, and then she does 4 treatments for 4 weeks. And she then does her visual tests that I showed before. And now here what we're doing is actually not just the light avoidance, the darken light setup, but we're also doing the optomotor again, so the drum, and try and see the visual acuity. And very nicely, the animals that receive the RD ones, untreated, so blind mice, don't respond to any visual stimulus here. The ones that receive the cells only have some capacity, but not great. But the ones that has received cells and electrical stimulation are now performing better than the ones that receive cells only. So we are starting to believe that actually electrical stimulation is a nice approach to enhance integration of the cells after transplantation. And we know it works in the spinal cord, right? We know electrical stimulation in the brain works for epilepsy, for example, and other neurological conditions. So it was an approach that we thought, okay, it might be worth testing. And she's done an amazing job. Ben already explained what. Now, with the project that Ben has been doing to try and generate a macula in the dish, what we want is actually to try later on and isolate that macula, so that cone-rich region, and transplant it at the back of the eye and see if we can replace the whole macula rather than just dissociated cones. And as I said before, now we're moving into GMP, so we want to produce these cells in a lab that can produce good enough cells that can go into humans, right? Everything that we do in the standard lab is fine and is great, but if you want to move into clinical trials, these cells need to go through a lot of quality control. And they actually need to be grown in a manufacturing facility that is absolutely controlled. And I have a big project ongoing to do that. And that's happening all over the place. So not a lot of labs working on it. We have some labs in the USA, of course, some in Japan. They actually have transplanted the first 3 patients with iPS-derived sheets of retinal organoids. So instead of dissociating and isolating just the cones, what they do, they cut the organoid in little pieces and they transplant that. Instead, it's a different approach. We go for dissociated. They prefer the whole sheets. And again, 3 patients have been injected. We have the group in the UK, so my ex-boss, doing that and hopefully going very soon into clinical trials. And we are doing it down here in Australia. So, yeah, that's it from me. That's the group. I was enjoying our Christmas party in the beach here. Thank you.